All right. Well, good afternoon, everyone. Thank you for joining this Iowa Learning Farms webinar. My name is Elena Whitaker. I'm the Water and Conservation Educator with Iowa Learning Farms. And today we're joined by Ellie Ellis from Colorado State University, and she's going to be talking about an across-the-fence comparison of soil carbon storage on 22 Iowa farms. Before I hand things over to Ellie, I just want to say, if you have any questions throughout the presentation, feel free to put them in the chat, and we will get them asked and answered at the end during our Q&A section. So with that, I'll go ahead and turn things over to you, Ellie.
All right. Thank you so much for inviting me. Excited to present some of the work from my PhD dissertation. I just defended my PhD at Colorado State, and most of my work took place in Iowa and southern Minnesota. So it's exciting to present this work to more of an Iowa audience. Today I'll be talking about some results from this on-farm study that I conducted in collaboration with 22 producers across Iowa and southern Minnesota. And the goal of the study was to measure soil carbon stocks on neighboring pairs of farmers that were using contrasting management systems. First, let's just remind ourselves how we've lost so much carbon from soil and how we can start getting it back to where it belongs. So CO2 is taken up from the atmosphere by plants through photosynthesis, and most of that carbon is used directly by the plant to grow roots and shoots, and some of it is released through roots to feed soil microbes. And as plants die and decompose, microbes decompose that organic material and release CO2 back into the atmosphere. So the cycle continues. And the organic matter that ends up residing in the soil as partially decomposed plant material and microbial byproducts, that organic matter contains a lot of carbon. So when carbon inputs to the system exceed carbon outputs, and that balance remains over an extended period of time, carbon sequestration occurs, or we start to rebuild the carbon that's been lost from soils. And in agricultural systems, carbon inputs come in the form of crop roots and root exudates, or in crop residues that are left on the field. And carbon can also be added in the form of manure, or compost, or mulch, or biochar. And carbon is lost to the system through outputs of microbial decomposition, which releases CO2, or erosion, which is carbon displacement from the system. So in native ecosystems, photosynthesis and respiration are mostly balanced. Those inputs equal outputs. But as we convert native ecosystems to agriculture by plowing up the land, soil organic matter and soil carbon is lost through decomposition, and CO2 is released into the atmosphere. Luckily, the use of conservation practices and restoration of native ecosystems, there's a fire alarm going off. One moment. Okay, sorry about that. So the use of conservation practices and restoration of native ecosystems are starting to send us in the right direction, but there's still a large depth of carbon in the soil. So we are at kind of a decision point here where we can continue to adopt practices that restore the balance of carbon, or we can degrade soils and continue to lose that soil organic matter. Luckily, we know there's a system of agriculture that can help replenish soil carbon without converting all agricultural land back to native ecosystems. So regenerative agriculture or conservation agriculture helps to increase carbon inputs to the soil and decrease carbon outputs by mimicking processes that occur in native ecosystems. And this is accomplished by adhering to 6 principles. The first is minimizing soil disturbance, which usually refers to no-till or reduced tillage. Keeping soil covered by eliminating periods of bare fallow, leaving crop residues on the field. Increasing diversity across the farm system. Integrating animals back into cropping systems whenever possible. keeping a living root in the soil for as much of the year as we can, and of course, designing these management systems with a specific context of the farm in mind. No one practice or suite of practices will work in every place. We know each of these practices has the potential to increase soil carbon at different rates and at different depths in the soil. So how do we know the potential of different practices to increase soil carbon? How do we measure these changes? Most of what we know about the potential to increase soil carbon comes from long-term experiments. So these are managed by universities around the country, like Iowa State, and across the globe. So in these long-term experiments, researchers will select usually a pretty uniform plot and establish block experiments where they measure the baseline soil carbon stocks and related properties. And then they'll implement a change in management, usually 1 or 2 practices at a time, and then measure the change in stocks over time. And while these experiments are invaluable, we know they couldn't possibly represent the full range of conditions encountered on real farms across the country. We'd need experiments on every corner of the country in order to do so. We know that most farms have many different soil types across the field. We know topography is often ununiform across a typical field, and that most farmers implementing conservation practices typically change more than one thing at a time. So experiments can't possibly represent that full range of conditions. We can use observational experiments conducted on real commercial farms to help fill this data gap and better represent the range of soil properties and management practices that characterize real farms. So I decided to conduct this study in the Midwest, coming from Colorado, because the Midwest is a perfect place to design an observational study of this kind, because there is so much farmland and so much regional variability in regenerative practice use. So this means that neighboring farmers located on similar soil types, similar topography, might be using very different management systems to grow the same crops. So I was able to harness that variability and find pairs of neighboring farmers across this region that were willing to participate in an across-the-fence comparison. So a little healthy neighborly competition for the sake of science. So I identified 11 pairs of neighboring farmers that were willing to participate in this study. One neighbor was using conventional management for at least 10 years, which I define as annual tillage and bare winter fallow. And the other neighbor was using a more regenerative system, which I define as at a minimum 7 years of no-till and 4 years of winter cover crops. Although many farmers in this study were using other practices or have used these practices for much longer. So how did I find farmers to participate in this study? Coming from Colorado to Iowa, a state that I don't live in, I spent an entire month in the summer of 2022 volunteering with Practical Farmers of Iowa and driving across the state, meeting with people, volunteering, attending field days, and just creating connections. And I'll say this was one of the most rewarding aspects of my work, and the time I spent really shaped the direction of my research. So once the 11 pairs were selected, I selected a specific field from each farm and used soil maps to select areas with similar soil types and topography between the two neighbors. Then in those areas of matching soil types, I randomly selected 9 sampling locations per field. And we buried a geolocator device at each of these sampling points so we can return in the future and see how things have changed over a more extended period of time. We collected 3 cores at each of these points and composited them across depths and sampled down to a meter. We also collected a crop litter sample or crop residue sample that we could see how crop residue differences played out between the farms. And this resulted in a total of over 1,700 samples, which kept us very busy in the lab. So since each of these regenerative farms had been using no-till and cover crops for differing numbers of years, we needed a simple way to compare them to each other. So I created this regenerative score, or regenerative index, that accounts for how many years of no-till and how many years of cover crops each of the farms had used. And here cover crops get a slightly higher weight because they have a higher potential increased soil carbon at all depths, or beyond just the surface. So let's dive into some data from these farms. This graph shows the difference in soil carbon stock between the regenerative and conventional neighbors. And it's organized by increasing regenerative score on the x-axis here. This is a composite score showing the number of years of practice use. So you can see the difference generally increases with the time practices have been used, but there are some unexpected results here too, where the conventional farm had higher carbon. We'll talk about those in a minute, but first, let's look at this field that had the highest regenerative score. This producer has been using a suite of regenerative practices for an extended period of time, and he supports other farmers in his community to do the same. Been no-till for about 30 years, cover crops for 20. It's also experimented with relay cropping and roller crimping. And also this farmer recently purchased the conventional field that we sampled. So this will be a really good one to monitor over time. Looking at this bar plot here, or the box plot here, you can see that the regenerative field, which is in the green, had higher soil carbon stocks at all depths, and significantly higher in the first two depth increments, which is about the first 12 inches. Let's go back to this plot here that shows all of the farms and talk about these three pairs in gold here, where the conventional farm had more soil carbon to a meter depth than the regenerative farm. And let's think about what could be driving that difference. When I saw these results, I thought back to what it was like to sample each of these farms and remembered that several of these fields were located on slight slopes. Nothing major, just it was a bit more driving up and down little hills to sample these fields. For the three pairs shown here, the regenerative field was located on a slight slope, while the conventional field was more or less flat. You can see that in these slopes listed here. The conventional field is on the left in each case, and the regenerative is on the right. So the regenerative fields were on like a 3 or 4 degree slope on average. So this might be a case where erosion over time has displaced a lot of soil carbon from these farms. And even with extended periods of regenerative practice use, it's hard to match a field where erosion was not as much of an issue. And it turned out that across all the fields that we sampled, the regenerative field was more likely to be located on a slight slope. And it makes sense that producers tend to adopt no-till and cover crops on their more eroded fields first. So for the next aspect of this work, I learned how to run a model called Descent that is used to predict soil carbon stocks by simulating the ecological processes that occur above and below ground. So Descent is a process-based model that requires a series of inputs for a farm or an ecosystem. So these inputs include climate properties, soil and field properties, and management events that occur on a daily basis. Descent then acquires a series of parameter files that were determined through years of experiments that tell us how these different input properties change outputs. So the outputs that the model gives us are plant growth or yield, soil carbon stocks, and greenhouse gas fluxes from the field. So most model development, most of the development that has gone into DayCent is carried out using data from controlled long-term experiments, which we already discussed before, don't reflect the range of conditions encountered on real farms. So my work aimed to test the model's ability to simulate processes occurring on real farms and simulate measured data. So here is the fit of the simulated soil carbon stocks compared to the measured soil carbon stocks. When I first ran the model, you can see here that the model is capturing about 27% of the variation in soil carbon. That's that weighted or squared. So yeah, that's not that great. We know the model performs much better when simulating data from controlled experiments. So it made me wonder if there was something I was missing when simulating these real world farms, something that wasn't being captured by the model. So I thought back to what we learned about erosion and that being a really important control on soil carbon on these farms. So I wondered if we could add erosion processes to the DayCent model. Erosion isn't thickly included in process-based models like DayCent, or it's not explicitly included. But we know erosion is a really important process in the Midwest, where over 30% of topsoil has been lost in some areas. So finding ways to add erosion into these models is really important. So I used an erosion model called Russell 2, which is the revised universal soil loss equation, to estimate yearly erosion rates on each of these fields based on soil properties and management data. And then I fed those erosion estimates into DayCent manually to see if it improved the estimations of soil carbon. And it turned out that adding erosion events significantly improved our estimations of erosion. It increased our R squared, which is the predictionability of the model, from 0.27 to 0.76. So a lot better. And just reminding ourselves that none of these fields were located on really extreme slopes or had experienced lots of erosion. It was all mild, but over 100-year period, that erosion really adds up. So it's important to simulate that over time. Here's a time series from one of those model estimations. This is that field that had been no-till for 30 years and cover crop for 20. This is the simulation of soil carbon stocks over a 40-year period here. You can see that the regenerative field in green was predicted to start slightly below the conventional neighbor shown in tan. That's based mostly on soil texture. And as this field adopts no-till and cover crops, shown by those two icons there, you can see the soil carbon really start to accrue. But we still don't quite match the measured data here in that triangle. So it's possible that DayCent can't really predict the impacts of practices like relay cropping and roller crimping that really increase biomass. And here's one of those pairs where there was a difference in slope. You can see in the simulation, the regenerative field that's located on like a 4-degree slope starts significantly below the conventional neighbor, but starts to accrue a lot of carbon once those practices are adopted. And here the simulation pretty well matches the measured values shown by the triangles. Here we're doing on time. Okay, got a few minutes left. So something else that we measured was soil organic matter pools. So this shows us where carbon is stored in the soil. if it's in more particulate form, where the organic matter is composed of fragmented plant materials or fragmented microbial materials, or if it's organic matter that's bound to mineral surfaces. So it might be bound to clay or silt-sized particles. We found that the regenerative fields that we sampled had more carbon in that mineral associated pool, which is, yeah, organic matter bound to clay and silt-sized particles. And we also found that the regenerative field had more carbon in that particulate organic matter pool. That's organic matter fragments that you can like see with the naked eye, looks like organic matter in the soil. Regenerative practices do seem to be increasing carbon in both of those pools, which is really, really good to know. And we think it's particularly the combination of no-till and cover crops that's leading to this increase, because the no-till decreases the outputs, the cover crops increase the inputs. So it's that combination that really seems to make a difference. So in summary, what did we learn from all this on-farm work? Many new insights were derived from these experiments. We learned that on-farm research can help expose overlooked variables or things that might not be as apparent in controlled experiments. We really highlighted the influence of erosion on slightly sloping fields. We learned that slope and erosion should really be added to process-based models of soil carbon. And we learned that a combination of no-till and cover crops really seems to build organic matter in these pools of stable carbon over time. So thank you all so much for listening, and I'm excited to answer any questions. Here's my email if you'd like to reach out with any specific questions too. So thank you.
Perfect. All right, Ellie, if you want to advance to the next slide, I'll go through some of those really quick before we do have some questions. And additionally, if people have more, keep submitting them to the chat. If you are joining the webinar today and would like to receive a CCA CEU, go ahead and email me, Elena Whitaker, at alenaw@iastate.edu by 5 o'clock today with the name you entered to watch the webinar if it's different than like your Zoom name, and your CCA number. And then next slide, please. If you are joining for the first time this year in 2025 to watch a webinar, if you want to submit this voluntary demographic survey, that would help us out a bunch. You can find it with the QR code. It's just a couple of questions, or at iowalearningfarms.org/survey. And then I think I've got a couple, like 2 more slides if you want to advance to those. Next week, join us again Wednesday at noon with Dr. Sylvie Broder. I'm not exactly for sure how to pronounce her last name. Talking about network meta-analysis quantifies benefits of 4R in management. So join that. She's going to be talking about some water quality and things like that. And then I think I've got one more slide here. If you want to watch this webinar or any other of our webinars, they are all recorded. You can find them on our website or also on our YouTube channel. So be sure to check those out. All right, and with that, we've definitely got some questions. So I'll get started asking you those. One person was wondering, in terms of when you were talking about your for your carbon versus regenerative score, they were wanting to know like the linear regression. And so you talked about that a little bit, but were the 3 outlier cases on the steeper slopes than the other? Were like they different?
Yeah, those 3 outlier cases were where the regenerative field was located on a slightly steeper slope. And yeah, when we designed the study, we thought we selected fields that were all on pretty moderate slopes or even flat land. But it just so happened when we looked back at those fields and did more of an analysis of the topography of each, that those pairs, the regenerative field was just slightly more uneven topography. So that was what we think was really the driver of those differences. Because yeah, with that plot, that bar plot that shows the regenerative score amongst all of those, we see that even a few of those pairs where the regenerative field had used practices for a long time or had a high regenerative score, if there was that slight slope difference between the pairs, that seemed to be the main driver. So I hope that answers it.
We got another question that's saying on the map where it showed like all of the paired farms, they said that the map indicated that there were few or maybe none of the farms were located on glacial till, high organic matter fields of north central and southern Minnesota. Did you have any of those fields that were on glacial till, high organic matter?
Yeah, I didn't have yeah, I tried to find a pair in that area. And just, who I connected with, there was nobody in that region. I did sample some long-term experiments managed by Iowa State that are located in that area. And definitely the organic matter and soil carbon's really high in that region. So yeah, if I'd sampled the field in that area, I would expect it to be really high. So it's basically like a historic wetland in a way.
Perfect. There's another question saying regarding the time series slide, they're wondering if you can put the amounts into context, like the 40 versus 60 milligrams of seed per hectare. And then basically they're wondering like what would the differences in the biomass outputs be?
Um, kind of, I'm assuming this is with the Day-Cent time series.
I think that's what they're meaning, yes.
Um, yeah, so like this here. Are you guys seeing my screen still?
No,
No.
we lost them.
I think it's not talking about this plot here. Um, so 40 versus 60 megagrams of carbon per hectare. What would the difference in the biomass outputs be? I think this is talking about the biomass output that we get is yield. So yeah, there was definitely an increase in yield over this time period. We calibrate the model to the reported yields. So yeah, over time, there's just more carbon going into the system, also more carbon going out. So that's something that's accounted for in this model here. I don't know if I can totally answer this question. Yeah, I
Yeah,
hope that maybe gets at it a bit. Yeah.
perfect.
And of course, there's just more biomass going in with the cover crop being added. So like I talked about in the beginning, it's all just carbon inputs versus carbon outputs that really drives that soil carbon stock.
Perfect. We got some other questions on were the differences, and I'm assuming they're meaning total soil organic matter statistically significant?
We didn't measure total soil organic matter. We measured total soil carbon.
Okay.
And they were statistically significant on some fields, but not on all. So yeah, in some cases, yes, some cases, no.
We've got another question on a little bit more about like the management. Basically, they're asking, were the no-tillers, the regenerative ag people still using anhydrous, or were there any differences like that between your conventional and regenerative fields?
Yeah, the fertilization practices kind of varied between the fields. That wasn't something we held constant. It was more just, what the producer tends to be doing. In general, the no-tillers were using less anhydrous, but some of them were still applying it as they had been before. So yeah, that was inconsistent throughout.
Okay, so do, do, do. So it talks about how they're talking about how you notice that most of the the carbon built up in the soil surface soil layer at the 0 to 30 under the regenerative practices, but they're wondering about that deep soil layer of the 30 to 100.
Yeah. Yeah, in general, the regenerative practices seem to be driving the difference in the top 30 centimeters. We did see differences in the deeper soil layer, but when we, kind of looked at other properties that were different between the soils, there was just more natural variation at depth. So it was harder to say if it was like management driving those differences or just natural variation in the parent material of the soil. So yeah, it might be making a difference, but could be more due to just natural variation too.
So a couple people are asking, can you comment on like the future of these fields? Like, do you think the carbon will continue to accrue, or that the regenerative ag fields might hit a plateau, or what do you think is going to happen in the future?
Yeah, that's a really good question. Yeah, it's definitely possible that they'll continue to accrue carbon, especially if these practices are used consistently over time. Um, but we know that soils do saturate in soil carbon. So a lot of that carbon is stored bound to mineral surfaces like clay and silt-sized particles can really bind and hold on to that organic matter. And once all those mineral surfaces are taken up, soils do have the potential to saturate. But there's still is potential to add more carbon in the form of the particulate organic matter. Um, so that yeah, that this study has kind of set us up to see what happens over time. I do hope I can go back to all these farms in about 10 years and see how things have changed, and that'll provide us a really, really good data set to see if that saturation is starting to occur.
Great. And that kind of leads into our next question. They're just kind of wondering if you could explain a little bit about the locator device that you put in all these fields, because yeah, it's hopefully going to last 10
Yeah,
years into the future and stay in the same spot. So,
totally. Yeah, we use these geolocator balls that are used to mark utility lines. It's like a little magnetic ball, it's like about this big, that we buried 1.5 meters in the soil, so well below any tillage depth. And yeah, hopefully they should stay in place. We've used these in other experiments, and they work great. So yeah, hopefully we can go back to the exact same spot on the field, but then like offset our sampling by about like 0.5 meter, so that we're not sampling the exact same spots, but it's like systematically resampled, so we can hopefully see a difference.
um, next person is wondering, so do you have, you talked a little bit about this earlier in a different question, but they're wondering about the yields for the corn and the soybeans. Like, do you have any observations and differences between the conventional and regenerative? And they're wondering along the lines of just because, um, especially for rented land, that the owner, and like the farmer, there might not be as much, just like a little bit of disconnect on why they want to be doing the regenerative if they don't own the land or things like that.
Yeah. Yeah, so I did collect 10 years of yield data from each of these farmers. And in some cases, those records were incomplete. So with the yield data that I did have, we did run a model of those yield to see if there was a difference between the conventional and the regenerative producers. And in our sample set, we found that there was no difference between the regenerative and the conventional. So yeah, it's a small data set to look at for that sort of thing, but our study shows that there's no difference between the two. And in terms of rented versus owned land, I'd say there's a pretty wide split between if these producers owned or rented their land. So that wasn't a driver we were really able to look at. But I will say there could have been a bit of a sampling bias in like just who I found to participate in this study. Usually I connected with the regenerative neighbor first, and then they connected me to a conventional neighbor. So I was just kind of, it wasn't a random sample, if that makes a difference.
Yeah. A couple people are asking, they would love to read like your full paper, your dissertation, like the published work. So where could people find that?
Yeah, the results from this aspect of the study are not out yet.
Okay.
They will be out soon.
Perfect.
I do have a paper out that just describes the sampling design and just like kind of lays the groundwork for how to design studies of this kind. I can share that paper. That's out in the Journal of Carbon Balance and Management, but I'll ask Elena how to maybe share that.
Yeah, perfect. Yeah. And then but the main date or information you're talking about today will be published soon, is what you're saying. Yes, perfect. Okay.
The final stage of the manuscript. Yep.
Perfect. Okay, another question is the total soil carbon accounting for differences in bulk density, or is this like a percent carbon on a mass to mass basis?
Totally. Yeah. Thanks for asking that question and clarifying. We did take bulk density samples in this case. We used a Giddings soil probe truck to take the samples. So we get just like whole cores where we can take the known volume of soil and the mass and get bulk density. So all the results that I presented here are in terms of soil carbon stocks, which is on a bulk density basis. Yeah.
We got a couple more questions. So if anyone still has a question, feel free to submit it. This person is wondering, does the descent predict the occurrence of saturation under the different or any type of management?
Yeah, the descent model will not necessarily predict saturation. The model works on these like theoretical soil carbon pools and doesn't really account for any sort of potential saturation. There are other soil carbon models that better account for those processes. But yeah, descent would not.
Okay. It looks like I got one more question here so far. Did you have any chance when you were sampling the soil to like study any of the soil microbes or anything like that?
Yes, we did take samples for soil microbial diversity. We looked at the bacterial diversity and fungal diversity between the fields. In 25 minutes, I couldn't present all the things that we did on these farms, but we did just try to collect as much information as we possibly could. And there are some really interesting results that have come out of that work that will be published as well. Generally saying that the soil microbial community is more diverse and interconnected on the regenerative fields. But yeah, those results will be out soon in a published paper.
Perfect. Someone is just asking if you could just in like a minute or two label a few of the bacteria or fungi that were of interest that you found or that were like unique or outstanding.
Yeah, I'm not a soil microbiologist, so remembering the exact names of species is a little difficult for me, but I know there were more arbuscular mycorrhizal fungi on the regenerative fields than on the conventional, so that's definitely of interest for water holding and nutrient cycling, and also just aggregate stability. So that's a pretty big one. And there did just seem to be like key differences in species composition between the two. Like there were unique communities between the two management systems. But yeah, we'll have to look at the paper for more details there.
So yeah, so basically, more info is coming. And people, you just got to keep Googling Ellie's name basically to find when those articles get or those papers get published. So
And I publish under the name Elizabeth. That's my full
Elizabeth,
name. So
okay.
Elizabeth Ellis is who to look for
Perfect.
there. So yeah.
Perfect. All right. So with that, I think that's all the questions I have. So thank you for presenting today. We had a ton of questions, a ton of like clapping hands coming across the Zoom and all that stuff. So I know people found this very interesting. Um, just a reminder for everyone else to join us next week with Dr. Sylvie Bridger from Purdue talking about 4R in management and things like that. And that's all. So thanks for joining us today, Dr. Ellis, now officially since you've uh, defended your thesis, your dissertation. And if you've got any last words, I'll let you finish out the webinar here.
Yeah, thank you guys all so much for listening. Yeah, I hope got something out of this and be on the lookout for my publications. So thank you.